Social immunity. Social immunity is any antiparasite defense mounted for the benefit of individuals other than the actor. For parasites, the frequent contact, high population density, and low genetic variability makes social groups of organisms a promising target for infection. This has driven the evolution of collective and cooperative anti-parasite mechanisms that both prevent the establishment of and reduce the damage of diseases among group members. Social immune mechanisms range from the prophylactic, such as burying beetles, smearing their carcasses with antimicrobials, or termites fumigating their nests with naphthalene, to the active defenses seen in the imprisoning of parasitic beetles by honeybees, or by the miniature hitchhiking leafcutter ants which travel on larger workers' leaves to fight off parasites. Whilst many specific social immune mechanisms had been studied in relative isolation, e.g., the collective medication of wood ants, it was not until Sylvia Cremer et al.'s 2007 paper Social Immunity that the topic was seriously considered. Empirical and theoretical work in social immunity continues to reveal not only new mechanisms of protection, but also implications for understanding of the evolution of group living and polyandry. Social immunity, also termed collective immunity, describes the additional level of disease protection arising in social groups from collective disease defenses performed either jointly or towards one another. These collective defenses complement the individual immunity of all group members and constitute an extra layer of protection at the group level, combining behavioral, physiological, and organizational adaptations. These defenses can be employed either prophylactically or on demand. Definition and definition. Sylvia Cremer defined social immunity in her seminal 2007 Current Biology paper Social Immunity as the collective action or altruistic behaviors of infected individuals that benefit the colony. She laid out a conceptual framework for the topic using examples from primates and eusocial insects. Cremer's definition focused on the collective benefits of behaviors and was adopted by other behavioral ecologists, e.g. Wilson Rich 2009, when describing immune phenomena which were contingent on the action of multiple individuals. Cremer went on to develop a series of comparisons between personal and social immune systems. She explained that her definition of social immunity encompassed the nature of these defenses, that they cannot be performed efficiently by C, but depend strictly on the cooperation of at least two individuals. However, in 2010, Sheena Cotter and Rebecca Kilner proposed to widen the definition of social immunity to any type of immune response that has been selected to increase the fitness of the challenged individual and one or more recipients, and recommended that the phenomena described by Cremer be known as collective immunity. This definition places importance on the evolutionary origin of behaviors rather than on their functional role at present. Cotter and Kilner explained that their broader definition would include immune behaviors in both animal families and social microbes, as well as situations where herd immunity exists due to investment in personal immunity, arguing that this allowed for investment. They further suggested that the evolution of social immunity be seen as one of the major transitions in evolution. Joel Munier proposed a further redefinition in his 2015 paper on the role of social immunity in the evolution of group living, suggesting that Cotter and Kilner's definition could problematically encompass immune defenses which arise not due to social life but due to shared location. Munier defines a social immune system as any collective and personal mechanisms. Upon exposure to a parasite, group members must both evaluate the threat it poses and the current level of colony infection in order to respond appropriately. Mechanisms of social immunity are often categorized by the stage of the parasite attack on a group of organisms they target. Some mechanisms are prophylactic, e.g. burying beetles, smearing their carcasses with antimicrobials, or termites fumigating their nests with naphthalene, whilst others are activated in response to a parasite challenge, e.g. imprisoning of parasitic beetles, by honeybees or by the miniature hitchhiking leafcutter ants who travel on larger workers' leaves to fight off. In insects, For a parasite to succeed in infecting multiple members of an insect group, it must complete three key tasks: be taken up from the extra nest environment into the nest, establish itself within the nest, multiply and spread to many more insect group members. Mechanisms of social immunity are thus often categorized by which steps they hinder and slash or block. Levels of sociality across the class Insecta range from eusocial species with cooperative brood care, overlapping generations within a colony of adults and a division of labor into reproductive and non-reproductive castes to solitary living, with many intermediate systems in between which, despite lacking full eusociality, still may exhibit parental care. Different systems of social organization alter both the possibility and cost-benefit ratio of social immune mechanisms, e.g. guarding the entrance to the nest requires both division of labor, whilst allogrooming merely requires behavioral interactions, though the absence of many behaviors currently only recorded in eusocial taxa from non-eusocial taxa may simply be due. For instance, it seems plausible that insects living in common nest sites could evolve to remove conspecific corpses from the nest or to isolate an infected group member, and yet these behaviors, and many more, have only been recorded. Alternatively, it may be the case that whilst the three conditions of eusociality themselves are not prerequisites for the emergence of these behaviors, secondary consequences of eusociality are. Perhaps the large number of individuals in eusocial colonies increases the efficiency of collective anti-parasite defenses, and thus their emergence begins to be selected for, or perhaps the preponderance of non-reproductive individuals is a necessary driver for the evolution of these behaviors, as when in a colony attacked by a parasite they can only increase their indirect. A lack of collective defenses in some eusocial taxa also shows that social immunity may also not always be adaptive due to life history costs or ineffectiveness against a particular parasite's infective strategy, and that living in a group does not necessitate the expression of any particular suite of social immunity mechanisms. For example, worker termites Pseudomopsis angusticalis do not discriminate between infected and uninfected conspecifics. Pharaoh and colonies Monomorium farianis choose to move into infected nests over uninfected ones, and queenwood ants Formica paralugubris are not repelled, but actually attracted to habitats contaminated with. Inhibiting parasite uptake into the nest. Inhibiting the parasite establishing itself within the nest. Once a parasite has entered the nest, colonies must now prevent the establishment of the parasite. This is particularly important for long-lived societies which without would accrue a high parasite load. In eusocial insects, the most common mechanisms to stop establishment involve sanitizing the nest and integrating substances with antimicrobial activity into nest material, nest hygiene behaviors. Examples include in the ant lineage, a unique antimicrobial, thoracic. Its acidic secretions have antibacterial and antifungal activity are deployed by ants to protect themselves, personal immunity, and other adults, as well as the vulnerable brood and nest substrate. It has been observed in leafcutter ants and weaver ants that broods raised by adults with non-functional antimicrobial glands are more susceptible. The Formosan subterranean termite Coptoderms formosanus fumigates its nest using naphthalene, known to humans as the main ingredient of traditional mothballs and antiseptic agents, to inhibit within-nest parasite. The wood-ant Formica paralugubris incorporates solidified conifer resin into its nest up to 7 of resin per liter of nest material, which inhibits the growth of bacteria and fungi. The terpenes in the conifer resin likely provide the antimicrobial activity, as they do in the antifungal secretions of soldier Castness Pseudoterms termites. The corpses of group members killed by an infectious disease present a serious hazard to colony health, as in natural environments. Death from disease is much more common than death from old age. One mechanism that has evolved to help neutralize this risk is necrophoresis, the removal of corpses from the nest by workers. Refuse piles or graveyards are common features of ant colonies, and workers remove corpses within the hour after death. Corpse deposition is biased towards downward slopes, which saves the corpse transporting worker energy as well as preventing any rain washing the corpses back into the colony. Necrophoretic behaviors are distinct from and responses to foreign inanimate objects as corpses are. By bringing corpses out of the nest, the ultraviolet radiation in sunlight can kill emerging fungal spores. Transport corpses to special refuse chambers rather than into the external environment. Temnothorax Liechtensteini ants remove the corpses of dead sisters as above, but bury newly dead foreign corpses inside the nest whilst one or a few individuals decide to transport or bury a corpse. Up to 25 workers are then involved in the undertaking of one corpse. Termites commonly bury and cannibalize corpses when C. formisanis colonies are infected with M. anisople. Necrophagy is common when the proportion of dead individuals in an area was low 15%, but as mortality increased then, so did burying behavior, up to 75%, 80% mortality above which carcasses were too numerous. Termites do occasionally remove dead from the nest, but this is uncommon. Leaving the nest at all is rare. Refuse chambers or piles/graveyards have not been observed. Research on undertaking behaviors in bees has concentrated on the honeybee Apis mellifera. 1–2% of a colony are undertaker bees, specialized for corpse removal. They inspect corpses with their antennae before rapidly dropping them from the nest. Waste material is removed continuously and deposited in refuse piles as above, or even by summitans into streams. The division of labor in Acromemex at Sanitary leafcutter ants includes individuals who work almost exclusively in waste management, waste material excreta, food refuse, etc. Frequently harbors Escovopsis, a microfungus that is parasitic on the fungus that Sanitary tends, as well as other parasites, so spatial separation. This is achieved by depositing waste material on downhill and downwind piles or in special deep underground refuse chambers. Distinct waste management workers also exist in Atacephalotes and Atacolumbica. The risk of waste worker ants contaminating the fungal garden/nest is lowered by other ants attacking them if they try to enter the nest. Whilst attacks are more frequent in species with internal refuse chambers, this also reinforces the division of labor. On the other hand, some species retain excreta and slash or anal exudates within the nest to utilize it for antiparasite purposes. C. formisanis employs its faces as building material for the nest carton. The use of fecal material promotes the growth of actinobacteria with natural antimicrobial activity, e.g. streptomyces, combating fungal parasites. Colony members of the buff-tailed bumblebee Bombus terrestris defend themselves against the virulent Cryphidia bombi parasite by consuming beneficial gut bacteria from the faces of conspecific after eclosing. Workers of the Cape honeybee Apis mellifera capsis socially encapsulate the parasitic small hive beetle Ethina tumida. A tumida feeds on the brood, pollen and honey, but due to its robust exoskeletal armor and habit of remaining motionless in small gaps with its head tucked turtle-like underneath its pronotum, honeybee workers find it difficult to decapitate and kill. Instead, workers seal the small hive beetles in using bee glue propolis. Construction of the propolis prisons by some workers takes 4 days, but is accompanied by other workers guarding the small hive beetle to prevent it escaping. Guards have been observed remaining day and night for up to 57 days and attacking the small hive beetle if it tried to escape. Some non-eusocial insects also sanitize their nests. The wood cockroach Cryptocercus punctulatus commonly defecates within the nest. This species nests in decaying wood, which often has a high microbe density, and feces have been found to have antifungal activity against them. Anisople, possibly mediated by microbes. The galleries constructed within spruce trees by spruce beetles Dendroctonus rufipnes are under threat from multiple species of fungus, which reduce spruce beetle fitness. Upon fungal invasion, adults begin secreting orally, and analysis of these secretions has revealed bacteria with antifungal activity. Fecal pellets are used to quarantine off fungally infested sections of the gallery. A waste management strategy exists in some group living and subsocial species. such as the subsocial Dugier's short-tailed cricket Anurogralis muticus. 5–10 minutes after defecating and returning to her eggs, A. muticus females return to the fecal pellets and removes them out of the chamber. Note: no other items are removed from the chamber. Upon discovering a carcass, subsocial N. vespuloids parents upregulate the antimicrobial activity of their anal exudates and smear them over the carcass, thus sanitizing the resource their brood will soon feast on. Inhibiting intragroup transmission. Other taxa. Social immune systems have been observed across a wide range of taxonomic groups. Allogrooming is found in many animals, for example, primates frequently groom others, a behavior which likely evolved for its hygienic function but has now been co-opted for its additional role in social bonding. Allogrooming in the common vampire, Bet Desmodus rotundus, is associated with the regurgitation of food and may allow other bats to identify which individuals are capable of supplying them with food. The allogrooming behaviors of horses and birds have also been studied. A range of sometimes elaborate cleaning symbioses also exist between many different species, especially in marine fish with their cleaning stations. Corsican blue tits Parus caeruleus prophylactically line their nest with aromatic plants such as Achillea ligustica, Helichrysum italicum, and Lavandula stoches to ward off mosquitoes and other blood-sucking ornithophilous bird-targeting insects. After the broader definition of social immunity by Cotter and Kilner, numerous examples of social immune behaviors within animal families can be given. Tungara frogs Engestomops pustulosus create foam nests during breeding in which embryogenesis occurs. These foam nests are imbued with ranaspumin proteins which provide defense against microbial attack. The three-spined stickleback Gasterosteus aculeatus, grass gobies Osterricesser ophiophalis, fringed darter Ethiostoma crossipterum, and two species of blenny also use chemical strategies. Intriguingly, microbes themselves have been found to have social immune systems. When a population of Staphylococcus aureus is infected with gentamicin, some individuals called small colony variants begin to respire anaerobically, lowering the pH of the environment and thus conferring resistance to the antibiotic to all other individuals, including those. An analogy can be drawn here with the social fever in bees described above. A subset of individuals in a population change their behavior and in doing so provide population-wide resistance. Using Richard Dawkins's concept of the extended phenotype, the healthcare systems developed by humans could be seen as a form of social immunity. Study species. The majority of studies on social immunity have been on eusocial insects. For example, Sylvia Cremer's work uses ants as a model system, whilst Rebecca Rosengaus works with termites. Outside of eusocial insects, one emerging model system is the burying beetle Necrophorus vespuloides. Necrophorus vespuloides. Evolution. Comparison with personal immunity. Many researchers have noticed marked parallels between the more familiar personal immune systems of individual organisms, e.g. T and B lymphocytes, and the social immune systems described above, and it is generally appreciated among ecological immunologists that rigorous comparative work between these two systems will increase of understanding of the evolution of social immunity. Whilst the specific physiological mechanisms by which immunity is produced differ sharply between the individual and society, it is thought that at a phenomenological level the principles of parasite threat and response are similar. Parasites must be detected rapidly. Responses should differ depending on the parasite in question. Spread of the infection must be limited, and different components of the individual. Cremer was the first to do this systematically and partitioned immunological phenomena into three categories: border defense, intake avoidance, soma defense, avoid establishment within non-reproductive components of an individual/society, and germline defense, avoid infection of the reproductive components of an individual/society. Example analogies from Cremer's paper are: border defense. Clotting in an individual's wounded blood vessels can be compared to the entrance plugging behavior of special end workers in response to a parasite attack. The circulatory system of an individual can be compared to nutrient distribution in social insect colonies, where a few forage and then distribute it to the rest of the nests. Cats and dog self-grooming can be compared to allogrooming. Soma defense. Granulome is formed to contain diseases that individual immune systems are struggling to eliminate. This can be compared to the social encapsulation seen against small hive beetles in honeybees. Apoptosis in response to disease can be compared to the killing of infected workers or the enforced suicide seen in temnothorax. Germline defense. The special protection afforded to reproductive organs, e.g. blood, ovary/blood testes barrier, increased number of immune cells relative to non-reproductive organs can be compared to the royal chamber found in social insects, where due to centrifugal polymorphism the queens, and sometimes kings, are cared for by young workers who have remained inside the nest their whole life. Transplant rejection caused by non-self major histocompatibility complexes is frequently thought to be a byproduct with no evolutionary function. However, Cremer cites cases such as the colonial star Ascidian Botrytis schlosseri, where recognizing foreign cells may have evolved as an adaptation. If so, then this could be analogous to the self-recognition systems. Specific immune cells in animals patrol tissues, looking for parasites, as do worker caste individuals in colonies. Cotter and Kilmer argue that not only is social immunity a useful concept to use when studying the major transitions in evolution, see below, that the origin of social immune systems might be considered a major transition itself. Role in the evolution of group living. The transition from solitary living to group living, identified by John Maynard Smith as one of the 7 major transitions in evolutionary history, brought with it many fitness benefits, increased anti-predator vigilance, foraging benefits, etc., and the opportunity to exploit a vast array of new ecological niches. But group living also has its pitfalls. Numerous studies have demonstrated an increase in contact-transmitted parasite load with group size increase, and thus research has been done on the role of social immunity in the evolution of early group living. Empirical evidence already exists from both interspecific and intraspecific comparative studies that an increase in population density drives an increase in personal immune effort density-dependent prophylaxis. However, there is also good evidence that the evolution of social immunity leads to a trade-off between effort into personal immune responses vs. effort in social immune responses. Physiological and genomic studies have shown that social conditions can lead to a reduction in personal immune effort. Personal immunity in the Australian plague locust Shortocetes terminifera decreases upon an increase in population density and increases when artificially isolated. Genomic studies reveal that infected solitary S. gregaria express more genes involved in immunity than infected individuals in the gregarious phase. Bombus terrestris workers also upregulate immune-related genes when experimentally isolated, and there are 3 times more immune-related gene families in solitary insects than in the eusocial hive. Joel Munier argued that the 2 seemingly contradictory relationships between personal immune effort and population density were a function of 2 assumptions implicit in the prediction that there should be a negative correlation. Group living is always associated with the expression of social immunity. This is false. Worker termites, see Angusticalis, do not discriminate between infected and uninfected conspecifics. Pharaoh and colonies Monomorium farianis choose to move into infected nests over uninfected ones, and queenwood and Formica paralugubris are not repelled, but actually. Social immune responses are costly for producers only in one species, Necrophorus vespiloides. Has this assumption been tested? A bacterial challenge to the larval resource led to a social immune response by the mother, and this response did lead to a reduction in lifetime reproductive success, i.e., there was a cost. Whilst advising that further studies in lots of different eusocial and non-eusocial taxa are required to better assess the validity of these assumptions, Munier notes that the existence of a trade-off between personal and social immunity could be masked or erroneously discovered in a population/species due to individual variation, e.g., low-quality individuals may not be able to assess what current knowledge of social immune systems suggested about whether social immunity was a byproduct or driver of complex group living. Munier delineated 30 different mechanisms of social immunity found in eusocial insects and looked for counterparts to these in prosocial and solitary insects, supporting the hypothesis that social immunity was a driver and not a byproduct of complex group living. 10 mechanisms had counterparts in prosocial insects and 4 in solitary species, though this does not imply that some mechanisms may evolve as a byproduct. Evidence that social immunity mechanisms are selected for at least somewhat due to collective benefits is lacking, though possibly due to the difficulty in isolating the immune benefits from the other benefits that social immunity. More studies on prosocial taxa would allow for phyletic analyses to recover the actual path of evolution that different mechanisms of social immunity took. Role in the evolution of polyandry. The origin of polyandry in nature and its adaptive value is a subject of ongoing controversy in evolutionary biology, partly due to the seemingly numerous costs it places on females' additional energetic and temporal allocation to reproduction, increased risk of predation, increased risk of sexually transmitted diseases, and increased risk of physical harm caused. One hypothesis for the evolution of polyandry draws on the disease resistance that increased genetic diversity supposedly brings for a group, and a growing body of evidence from insect taxa supports this hypothesis, some of it discussed above. Concept. Social immunity is the evolution of an additional level of immunity in the colonies of eusocial insects, some bees and wasps, all ants and termites. Social immunity includes collective disease defenses in other stable societies, including those of primates, and has also been broadened to include other social interactions, such as parental care. It is a recently developed concept. Social immunity provides an integrated approach for the study of disease dynamics in societies, combining both the behavior and physiology, including molecular-level processes of all group members and their social interactions. It thereby links the fields of social evolution and ecological immunology. Social immunity also affects epidemiology, as it can impact both the course of an infection at the individual level as well as the spread of disease within the group. Social immunity differs from similar phenomena that can occur in groups that are not truly social, e.g., herding animals. These include identity-dependent prophylaxis, which is the upregulation of the individual immunity of group members under temporal crowding, and Roman II herd immunity, which is the protection of susceptible individuals in an otherwise immune group, where pathogens are unable to spread due to the high ratio of immune to susceptible hosts. Further, although social immunity can be achieved through behavioral, physiological, or organizational defenses, these components are not mutually exclusive and often overlap. For example, organizational defenses, such as an altered interaction network that influences disease spread, emerge from chemical and behavioral processes. Disease risk in social groups. Sociality, although a very successful way of life, is thought to increase the per-individual risk of acquiring disease simply because close contact with conspecifics is a key transmission route for infectious diseases. As social organisms are often densely aggregated and exhibit high levels of interaction, pathogens can more easily spread from infectious to susceptible individuals. The intimate interactions often found in social insects, such as the sharing of food through regurgitation, are further possible routes of pathogen transmission. As the members of social groups are typically closely related, they are more likely to be susceptible to the same pathogens. This effect is compounded when overlapping generations are present, such as in social insect colonies and primate groups, which facilitates the horizontal transmission of pathogens from the older generation to the next. In the case of species that live in nests/burrows, stable, homeostatic temperatures and humidity may create ideal conditions for pathogen growth. Disease risk is further affected by the ecology. For example, many social insects nest and forage in habitats that are rich in pathogens, such as soil or rotting wood, exposing them to a plethora of microparasites, e.g. fungi, bacteria, viruses, and macroparasites, e.g. mites and nematodes. In addition, shared food resources, such as flowers, can act as disease hubs for social insect pollinators, promoting both interspecific and intraspecific pathogen transmission. This may be a contributing factor in the spread of emergent infectious diseases in bees. All of these factors combined can therefore contribute to rapid disease spread following an outbreak, and if transmission is not controlled, an epizootic and animal epidemic may result. Hence, social immunity has evolved to reduce and mitigate this risk. Components of social immunity in insect societies. Nest hygiene. Social insects have evolved an array of sanitary behaviors to keep their nests clean, thereby reducing the probability of parasite establishment and spread within the colony. Such behaviors can be employed either prophylactically or actively upon demand. For example, social insects can incorporate materials with antimicrobial properties into their nest, such as conifer resin or fecal pellets that contain symbiont-derived antimicrobials. These materials reduce the growth and density of many detrimental bacteria and fungi. Antimicrobial substances can also be self-produced. Secretions from the metapleural glands of ants and volatile chemical components produced by termites have been shown to inhibit fungal germination and growth. Another important component of nest hygiene is waste management, which involves strict spatial separation of clean nest areas and waste dumps. Social insect colonies often deposit their waste outside of the nest, or in special compartments, including waste chambers for food leftovers, toilets for defecation, and graveyards, where dead individuals are deposited, reducing the probability of parasite transmission from potentially infected cadavers. Where social insects place their waste is also important. For example, leaf-cutting ants living in xeric conditions deposit their waste outside the nest, whilst species living in the tropics tend to keep it in special chambers within the nest. It has been proposed that this difference is related to the likelihood that the external environment reduces or enhances microbial growth. For xeric living ants, placing waste outside will tend to inhibit infectious material, as microbes are usually killed under hot, dry conditions. On the other hand, placing waste into warm, humid environments will promote microbial growth and disease transmission, so it may be safer for ants living in the tropics to contain their waste within the nest. Honeybees have evolved the ability to actively maintain a constant temperature within their hives to ensure optimal brood development. Upon exposure to Ascosphaerapes, a heat-sensitive fungal pathogen that causes chalk brood, honeybees increase the temperature of the brood combs, thereby creating conditions that disfavor the growth of the pathogen. This social fever is performed before symptoms of the disease are expressed and can therefore be viewed as a preventative measure to avoid chalk brood outbreaks in the colony. Sanitary care of group members. Sanitary care reduces the risk of infection for group members and can slow the course of disease. For example, grooming is the first line of defense against externally infected pathogens such as entomopathogenic fungi, whose infectious canidia can be mechanically removed through self- and allogrooming social grooming to prevent infection. As canidia of such fungi only loosely attach to the cuticle of the host to begin with, grooming can dramatically reduce the number of infective stages. Although grooming is also performed often in the absence of a pathogen, it is an adaptive response, with both the frequency and duration of grooming self and allo increasing when pathogen exposure occurs. In several species of social insect, allogrooming of contaminated workers has been shown to dramatically improve survival compared to single workers that can only conduct self-grooming. In the case of ants, Pathogens large enough to be removed by grooming are first collected into the infrabuccal pocket found in the mouth, which prevents the pathogens entering the digestive system. In the pocket, they may be mixed labial gland secretions or with poison the ants have taken up into their mouths. These compounds reduce germination viability, rendering Canidia non-infectious, when later expelled as an infrabuccal pellet. In the case of termites, pathogens removed during grooming are not filtered out before entering the gut, but are allowed to pass through the digestive tract. Symbiotic microorganisms in the hindgut of the termite are also able to deactivate pathogens, rendering them non-infectious when they are excreted. In addition to grooming, social insects can apply host and symbiont-derived antimicrobial compounds to themselves and each other to inhibit pathogen growth or germination. In ants, the application of antimicrobials is often performed in conjunction with grooming to provide simultaneous mechanical removal and chemical treatment of pathogens. In ants, poison can be taken up into the mouth from the acidopore, the exit of the poison-producing gland at the tip of the abdomen, and stored in the mouth. In the Antlachius neglectus, the poison produced by the acidopore is composed largely of formic acid 60%, but also contains acetic acid 2%. Inhibition assays of the poison droplet against the fungal pathogen metarrhizine found that the formic acid alone substantially reduces fungal Canidia viability, but that all poison components work synergistically to inhibit Canidia viability by as much as 96%. Dealing with infected group members. Infected individuals and diseased corpses pose a particular risk for social insects because they can act as a source of infection for the rest of the colony. As mentioned above, dead nestmates are typically removed from the nest to reduce the potential risk of disease transmission. Infected or not, ants that are close to death can also voluntarily remove themselves from the colony to limit this risk. Honeybees can reduce social interactions with infected nestmates, actively drag them out of the hive, and may bar them from entering at all. Hygienic behavior is the specific removal of infected brood from the colony and has been reported in both honeybees and ants. In honeybees, colonies have been artificially selected to perform this behavior faster. These hygienic hives have improved recovery rates following brood infections, as the earlier infected brood is removed, the less likely it is to have become contagious already. Cannibalism of infected nestmates is an effective behavior in termites, as ingested infectious material is destroyed by antimicrobial enzymes present in their guts. These enzymes function by breaking down the cell walls of pathogenic fungi, for example, and are produced both by the termite itself and their gut microbiota. If there are too many corpses to cannibalize, termites bury them in the nest instead. Like removal in ants and bees, this isolates the corpses to contain the pathogen, but does not prevent their replication. Some fungal pathogens, e.g. Ophiocordyceps, Pandora manipulate their and hosts into leaving the nest and climbing plant stems surrounding the colony. There, attached to the stem, they die and rain down new spores onto healthy foragers. To combat these fungi, healthy ants actively search for corpses on plant stems and attempt to remove them before they can release their spores. Colony-level immunization. Immunization is a reduced susceptibility to a parasite upon secondary exposure to the same parasite. The past decade has revealed that immunization occurs in invertebrates and is active against a wide range of parasites. It occurs in two forms: I-specific immune priming particular parasite, or Roman II, a general immune upregulation that promotes unspecific protection against a broad range of parasites. In any case, the underlying mechanisms of immunization in invertebrates are still mostly elusive. In social animals, immunization is not restricted to the level of the individual, but can also occur at the society level via social immunization. Social immunization occurs when some proportion of the group's members are exposed to a parasite, which then leads to the protection of the whole group upon secondary contact to the same parasite. Social immunization has been so far described in a dampwood termite fungus system, a garden ant fungus system, and a carpenter and bacterium system. In all cases, social contact with pathogen-exposed individuals promoted reduced susceptibility and their nestmates increased survival upon subsequent exposure to the same pathogen. In the ant fungus and termite fungus systems, social immunization was shown to be caused by the transfer of fungal conidia during allogrooming from the exposed insects to nestmates performing grooming. This contamination resulted in low-level infections of the fungus in the nestmates, which stimulated their immune system and protected them against subsequent lethal exposures to the same pathogen. This method of immunization parallels variolation, an early form of human vaccination, which used live pathogens to protect patients against, for example, smallpox. Organizational defense. Organizational disease defense or organizational immunity refers to patterns of social interactions which could, hypothetically, mitigate disease transmission in a social group. As disease transmission occurs through social interactions, changes in the type and frequency of these interactions are expected to modulate disease spread. Organizational immunity is predicted to have both a constitutive and an induced component. The innate, organizational substructure of social insect colonies may provide constitutional protection of the most valuable colony members, the queens and brood, as disease will be contained within subgroups. Social insect colonies are segregated into worker groups that experience different disease hazards, where the young and reproductive individuals interact minimally with the workers performing the tasks with higher disease risk, e.g. foragers. This segregation can arise as a result of the physical properties of the nest or the differences in space usage of the individuals. It can also result from age or task-biased interactions. Distinct activity patterns between group members, e.g. individuals with relatively higher number of interactions or high number of interaction partners, has also been hypothesized to influence disease spread. It is further assumed that social insects may further modulate their interaction networks upon disease coming into the colony. However, the organizational immunity hypothesis is currently mainly supported by theoretical models and awaits empirical testing.